turn on your camera and mic and join me on the stage for walking us through our new CAN disease modeling platform. Something that is also really exciting is walking us through how we started with a resource that was isolated from patients, and we are now seeing very mutation-specific effects. We're learning more on the cellular level about can impact cellular function. And we keep referring to this platform as a disease modeling platform, which means that there are going to be certain clinical phenotypes that are trying to be modeled or represented in these cells in a dish. So can you tell us a little bit more, clarify the specific clinical phenotypes that these assays are trying to represent and that we can learn from?
Well, well, thanks, Tom. The MEA phenotypes, the hyperexcitability phenotypes, is a surrogate for the seizure-genic phenotypes that Wendy Chung described that a number of CAN patients exhibit. And I think the axonal outgrowth phenotype that we observe may parallel some of the neurodevelopmental delays. So those are the two obvious phenotypes that might translate to what you see in the clinic. The asynchronous calcium and then the Neucyte expression of KIF1A in the neurites relative to the soma. Those are more biologically related functions, and then may not necessarily, we still do not understand how those impairments in biology might manifest clinically.
Great. I see a question from Audrey in the chat. Well, I do want to point out we've got a handful of great questions in the chat, some that skew more towards general therapeutic development questions that we will address after the conference as well. So everyone's questions will get answered. But this one is from Audrey. It says, nice talk, thanks. Is there a phenotype common to all different mutations in iPSCs that you've tested or worked with?
So far, I think the MEA hyperexcitability is the one that's been common to the two that we examined. We examined the E253K mutation and then the P305L. These were the two lines that we had access to, and I know that the KIF1A.org group has been working with JAX.org to develop a library of mutations across within a single background, so having isogenic model. And I think that this will be available to the KIF1A.org research network shortly.
That's a great point of distinction. Thank you, Wayne. While we started with 2 mutations, the overall goal is to expand this platform to many more KIF1A mutations as well. Now, I dropped a little tidbit in my presentation about some potential compound screening on the horizon with this platform at Neucyte. Very generally, very broadly, could we discuss sort of the themes of these compounds that are going to be initially tested in this first batch of screening?
I think that the themes of some of these compounds initially will target, again, axonal outgrowth phenotypes, and then some of the compounds that we commonly see that are in the clinic that address seizures. We want to see how they affect the hyperexcitability phenotype, and then because we have a platform with both 2 different types of neurons, excitatory and inhibitory, we'll be testing compounds that can affect inhibitory activity.
Great. I like this one. This is kind of a thought question. KIF1A variants are non-hotspot. Any ideas to develop a functional assay that can classify all new KIF1A pathogenic variants to provide insight in prognosis or extent of disease, maybe using CRISPR? What are your thoughts?
That is I don't, it came from anonymous. That is a great suggestion. There are a number of labs globally looking to utilize the novelty of the CRISPR-Cas9 system to develop these type of like agnostic screens, and then in the case of KIF1A, it would be I think a pretty a straightforward approach to try to introduce randomly through CRISPR-Cas9 mutations throughout the protein, and then to identify which ones affect what type of phenotypes. That is a great idea.
Absolutely. Next one, do you specifically need patient fibroblasts to do this work, or could you simply generate pathogenic variants? I'm assuming they're referring to in a specific cell line.
Correct. I think there are pros and cons to both approaches. I think what we still don't know, because there are so many different variants. We don't understand how they can manifest in some pretty different clinical outcomes. And so I think, are there other patient genetic backgrounds that play a role when you inherit some of these de novo mutations? That I don't know. And so then if you used a wild type line and then introduced all these pathogenic variants, that would maybe get at common mechanisms versus divergent mechanisms, since you're using the same genetic background. So they each have their pluses and minuses. And so we've currently started on the one where we've looked at patient specific mutations.
Absolutely. And to address this question as well, we recognize, like Wayne said, the utility, pros and cons of both types of scenarios that he's described. And so we do have both patient-derived cell lines as well as genetically engineered cell lines that can be used in these therapeutic development projects to help get at the questions from different angles as needed. I just saw a question from Heidi. Where'd it go? There it is. Is an evoked PTX-induced seizure-like event more likely to favor compounds with specific mechanisms of action?
The answer there is yes. And so in addition to PTX, we're going to be exploring other compounds, and also looking at, over time, whether or not at baseline we can also detect some hyperexcitability phenotypes. And so we're working with other groups who have platforms such as high density microelectroarray, which would allow us to look at things like I alluded to in my pre-recorded presentation about axonal initial segments, and the role that KIF1A plays in establishing that, and how that regulates action potentials, in which impairments in the AIS will lead to hyperexcitability. So yes, I do think that with the PTX model, we are going to select for certain compounds, but we are exploring other phenotypes that will cover the spectrum, we believe.
Great. And one last question, sort of taking a big picture, from your perspective, what is the most exciting finding that your team has uncovered since working with these KIF1A patient-derived cell lines?
I think that the hyperexcitability phenotype has been the most exciting, because it's been very robust, and then it allows us to utilize our in-house high throughput platform to potentially screen compounds very rapidly. And
Excellent.
so that's really it really will allow us to accelerate the drug discovery process.
I'm so excited. We can't wait for our next update. Dr. Wayne Poon, thank you so much for joining us and for your presentation. I extend our thanks to the team at Neucyte for your collaboration and effort on our CAN disease modeling and drug screening platform.